Ladies and gentlemen, welcome, and thank you for joining us today to discuss SanBio's clinical programs. Also, I wanted to thank ARM organization to have given us this opportunity. This is our disclaimer. SanBio is a company focused on regenerative medicine. The campuses are on Mountain View, California, and Tokyo, Japan. The main focus of the company is on chronic neurologic diseases. And the program which is the most advanced is in TBI, or traumatic brain injury. Basically, the therapy, which is called SB623, and I'll explain in a minute as what they are, targets the patients who have chronic motor deficiency after an old traumatic brain injury. These patients do not have any therapeutic option to change biologically their brain. They have, however, physical therapy and rehabilitation that occurs on and on during their life. Now, this program has had a phase 2 trial, which was successful in showing improvement of motor function in those patients. And right now, we are preparing a biological license application for Japan, regulatory, in context of a designation called Sakigake, which allows us to do that to submit in earlier stages, and I will explain later on. We are also, parallel to that, of course, preparing for our commercial launch because we are expecting that the approval from Japan allow us to commercialize the product. We have also other programs in chronic neurological diseases, such as in stroke, and that includes ischemic stroke and hemorrhagic stroke. We have also a rich pipeline in preclinical, which we are advancing into later stages in order to be in clinical in the future. Now, this is our pipeline. As you can see on the top, the traumatic brain injury in Japan program is moving forward to submission. And then in the US and Europe, we are planning a phase 3 trial. And we have been discussing with the FDA in preliminary discussions in order to plan for those trials. The ischemic stroke and hemorrhagic stroke we'll be discussing with PMDA, the regulatory agency in Japan, in order to moving on to either a 2B phase or a phase 3 trial as well. We have also a retinal disease program, and that program has been partnered with Ocumension. We have some other products and other programs as well, as you can see, that they are in preclinical stage. And I explained that our purpose is to advance those preclinical to clinical in later times. Now, when you look at the chronic brain injury, that would be in traumatic brain injury or consequent to ischemic stroke or hemorrhagic stroke, there is a very important unmet need across the world. In every continent, all the big countries, you can see the numbers here. For example, in US, they are in the millions. Now, SB623 is a mesenchymal stem cells that has been modified by a transient transfection of Notch1. And I will explain to you in a minute the mechanism of action. The product is produced through an allogenic process. And as you know, the allogenic process can allow you to have mass production and have off-the-shelf product that you can use anytime for patients, as opposed to autologous, in which the patient's cells are modified and given back to the same patient. So every patient is his own or her donor. Now, how the SB623 works. We have studied that product over a long time, and we have seen in the injured brain, there is increase in neuropoiesis, angiogenesis, and the blood-brain restoration, as well as immunomodulation. And these are relatively related to each other as well. For example, the angiogenesis and blood-brain barrier repair basically brings more nutriment and oxygen, but at the same time prevents the inflammatory cells to infiltrate the brain, and therefore you're going to have an immunomodulatory effect. You have also a conditioning of the brain microenvironment, new synapses, as well as remyelinization of the axons. Now, how this drug is given to the patient. The drug is injected through stereotactic neurosurgery into the surrounding area of the lesion. And we see in preclinical, in vivo, that there is activity there, and those are the mechanisms of action we explained. And in human, we have seen improvement of motor impairment as expected. Now, our program in traumatic brain injury, the phase 2 trial consisted of enrolling 61 patients with had the motor deficit, which were related to a brain area identified on MRI. The patients were randomized in 4 different groups: sham surgery group and 3 dose group, 2.5, 5, and 10 million cells. The patients were followed for over time, and the primary endpoint was assessed at 6 months. The primary endpoint was the improvement from the baseline of Fugl-Meyer motor scale, which has been, and you're familiar with, has been used often in the clinical trials. Now, what was the result? The improvement of Fugl-Meyer motor scale over time was increased in significantly in the treatment group. And the difference, the delta with the control group, it increased as well. So that at 6 months, there was an improvement of 8.3 points in the treatment group, the pooled SB623 group, compared to 2.3 in the control group, with a p-value of 0.04. Now, this trial, of course, had other endpoints as well. There were disability rating scale, action research arm test or AROT, gait velocity, global rating of perceived change. Majority of those improved as well. And then there was no significant difference in the rate of adverse event between the treatment groups and the control group. There was no dose-limiting toxicity or deaths, or no patient withdrew due to the adverse event. There was no relationship between the dose and the frequency of adverse events either. The study was published in Journal of Neurology in January 2021. Now, where are we on our biologic license applications to PMDA in Japan? This is as you may know, the Sakigake designation is a privileged path for regenerative medicine in Japan for being approved with the data, which may be phase 2 data. And that's the path we are using. And that includes a period of consultation with the PMDA, who is extremely important and useful in order to have a successful submission. And that's exactly what we're doing right now. And in ischemic stroke and hemorrhagic stroke, we will be discussing with PMDA about our next step of phase 2B or phase 3 trial as we go along. Now, this is an illustration of the Sakigake path to conditional approval. That submission will allow you a conditional approval and market launch. And that condition is to have another post-marketing study later on. But meanwhile, you can be on the market. Now, of course, speaking of the market, we are preparing for it, and that would include the strategic launch planning by creating promotional material and regulatory documentation that we need, collaborate with the sites, neurosurgeons, the KOLs, and different facilities, building the logistics, which is needed to patient registration product, and of course, determination of the pricing discussion with the authorities, and building, of course, a sales structure. The target healthcare for that would be multiple, including in that list on this slide. To summarize, the SB623 is a modified mesenchymal stem cell. That product has had shown in preclinical and clinical a significant efficacy on people with chronic brain injury and motor deficit. So the purpose here is to improve those motor deficits so that these people have a brain which has regenerated, repaired, or revitalized so they can continue to have a better life. An early and near stage is a submission and aiming at approval in Japan, and then commercialize, of course, and then go on as well into the stroke. That would be the hemorrhagic stroke or ischemic stroke, and continue to strengthen our pipelines so that we can move on with other indications and other products that we have into later stages. Thank you very much. I appreciate your time.